Hi, everyone. Welcome to this semester's final lecture. We're going to talk today about learning and memory, and this is going to complete our four chapters for the final midterm, which you'll start on Friday and have some period to complete. Today, we're going to talk about 5 topics. First, we're going to consider the overall idea of learning and memory and how they relate to the other topics that we've covered in the course. We'll dissociate the various types of memory first before we zoom in on various circuits that underlie our explicit and our implicit memory. Then we'll finalize by talking about brain plasticity and some of the changes that can take place in our brain that leads to these long-term changes. And finally, we'll end with recovery from brain injury. The brain is inherently plastic, as you know, and our experiences change the activity of our brain and also the physiology of our brain. Our development, our culture, our preferences, and the way we cope with our lives can all affect our brain. Learning is common across these experiences, and so the effect of the environment on our brain is for it to learn and adapt. So neuroplasticity we can define as the nervous system potential for physical or chemical change, and this enhances its adaptability to the environment. And that's one of the goals of our brains, of course, is to adapt. Learning we can define as a change in an organism's behavior as a result of something that's occurred to them, as some experience. And a corollary of learning, of memory, is some ability to recall or recognize a previous experience. And this relates to the idea of a memory trace. A memory trace is some mental representation of that physical experience, or of that experience, which we assume to be some physical change or activity in the brain. It most likely involves some change in the synaptic activity. You've studied in your classes about different types of learning, I believe, and one we can refer to as Pavlovian conditioning or Pavlovian learning. These are learning between a conditioned response and an unconditioned stimulus. So the classic example is a dog learning to salivate when it hears the bell that's associated with its food, because it comes to learn that the bell leads to food. There's a couple terminologies that you'll recall in this topic, the conditioned and stimulus and conditioned response, and the unconditioned stimulus and the unconditioned response we'll go over. So Pavlovian conditioning is a procedure whereby a neutral stimulus, like a tone, which we call the conditioned stimulus because it's being conditioned, comes to elicit a response like salivation, called the conditioned response, because it's repeatedly paired with something in the world, such as the delivery of food, which is an unconditioned stimulus, doesn't need any conditioning, and it naturally leads to an unconditioned response, salivation. This is also called classical conditioning or respondent conditioning. And so here you can even write it as an equation, the CS, the conditioned stimulus, plus the unconditioned stimulus, normally at first evokes an unconditioned response because of that unconditioned stimulus. After several pairings, the conditioned stimulus itself will elicit a conditioned response that's similar to that unconditioned response. So the conditioned stimulus is the neutral stimulus that triggers the conditioned response after it becomes associated. The unconditioned stimulus is a stimulus that unconditionally, either automatically or naturally, creates this response, like the food in the mouth of the dog. The unconditioned response is this naturally occurring response to that unconditioned stimulus. In this case, it's salivation when there's food in the mouth. And the conditioned response is this acquired behavior that the animal obtains through the learning. They learn to create this unconditioned response in response to the conditioned stimulus. An example that we can use in humans is called eye blink conditioning, which is actually an aversive type of conditioning, and that generally doesn't feel pleasant. In this case, a tone, the conditioned stimulus, is associated with a painless but unpleasant puff of air to the eye called the unconditioned stimulus. Normally, people blink when they get a puff of air to their eye. This is the unconditioned response, but over time, learning occurs, and the conditioned stimulus, the tone, comes to elicit that blink, irrespective of the air being puffed, as you might have experienced at the optometrist on the second eye, where they go to measure your ocular pressure and they shoot a puff of air into your eye. You might be more prepared for the timing of that one and flinch before it happens. In contrast to this classical conditioning or respondent conditioning, we can think of another type of learning called operant conditioning, which involves the animal operating in their environment and looking at the consequences of their behavior and increasing the probability of doing those behaviors that lead to good consequences. So Thorndike, over 100 years ago, originally described a learning procedure in which animals were placed in boxes and had to solve the kind of the trap of the box to get out, like the escape rooms that people enjoy these days. And so escape rooms are a great example of this operant conditioning. Of course, as we, if we do it a second time, we'll be much faster at learning to leave the room. So the example of operant conditioning, or you can think of this as similar to the type of learning that Skinner did in Skinner boxes and the behaviorist set of research that followed. An animal is placed in a box with a number of contraptions, one of which will open the box and allow it to eat food. Originally, the cat just kind of wanders around and randomly tries stuff until it accidentally presses one of the levers that leads to the door opening, and it quickly gets a reward for that and learns to perform that behavior more in the future. And so it tends to repeat this behavior that leads to reward, and it tends to decrease behaviors that lead to punishment. So this has led to this field of learning called reinforcement learning as well, which is now popular in computing science. We can further distinguish our memory along a different lines, in the lines of implicit and explicit memories, which is something that you've thought about. This is the difference between riding a bike and telling your teacher what the components of the basal ganglia are. So implicit memory is an unconscious memory where we can demonstrate our knowledge through some sort of skill. But you can't explicitly retrieve the information, and you can't tell someone how to do it usually. Whereas an explicit memory is conscious, and you can retrieve the item and you can indicate if you have heard this before, and you know when you've said the correct answer. So most of your exams in biology are usually examples of this explicit memory for the facts, whereas something like math, where you have to learn a procedure to do a particular type of operation, is something more similar to implicit memory. Some other examples of implicit memory are this Gollum figure test. This is an interesting example where participants are given increasingly clear pictures of an object in noise, and they have to identify the object as soon as possible. And over time, people learn to identify the object with less and less of the image shown. And so in a week, if you show people the same objects, they'll be able to identify this as a tiger earlier in the series than when they did the first time. We'll learn today about patients that have a disorder called amnesia, that have an inability to create new memories. And even though they're not able to explicitly recall having tried this task ever, they too are able to improve at the task over time, showing there's this differentiation between the type of memory that they're unable to perform now, that they're amnesic, and this implicit memory that they still have intact. Another example of implicit memory is the pursuit rotor task, in which this rotating disc and target are spinning around, and the participant has to learn to track it. Slowly over time, one can get better at this after practicing each day. And even amnesic patients that don't recall ever having tried the task also perform better over time. Another way of thinking about this distinction between implicit and explicit memory is declarative versus procedural memory. So a declarative memory is a more explicit memory that we can recount. We can detail the time and place of the event, and it's something that often is lost in amnesia. Whereas a procedural memory is a particular movement sequence or how to perform some behavior, like to sing a song. So from a practical point of view, these are not very different from the implicit-explicit distinction we had on the previous slides. And this distinction between explicit and implicit has, this dichotomy has many labels that you can look in this chart and think about. And things like fact versus skill, or memory versus habit, knowing that versus knowing how. So one of them is about recalling specific information, and the other is knowledge that we're not aware of. And you can see that Pavlovian conditioning and operant conditioning are also included in this. They're both types of implicit memory that people aren't explicitly aware of. The first step in memory is often thought of as encoding. We have to take the incoming sensory information and turn it into some sort of code that our brain understands. So we take a bunch of activity of neurons of our retina and code it into the color red, for instance, so that we recall the color of something tomorrow. This can be done either implicitly from the bottom up, so you could encode things the way that you perceive them, or you can think of encoding from a top-down way. Say when the textbook gives an example of looking for your keys. When you go around the house looking for your keys, the stimulus that you're taking in and how you process them are much different than when you're just wandering around looking aimlessly. And so there's some organization of the information prior to encoding in this explicit task. So in implicit encoding, the person's more passive, whereas when we're doing explicit encoding, we have some sort of active role. You come to class and you're learning about the topics of class and the framework of the knowledge you've learned in the rest of the class. One example of implicit learning is something called priming. Priming is a very common cognitive effect in which we can impact people's responses to a stimulus just by kind of cueing them with something earlier on. And the textbook gives examples of if you show a word like, if you present a list of words that all are similar to seasons, and you present a list of words that are all similar to activities that humans can do, and then you ask people to define the word fall, people primed with the list of words that had a lot of seasons in them will say that fall means one of the seasons of the year. And people prime with a list of words associated with different activities humans can do will say that fall means someone tumbling over. So you can think of this priming as this very effective way of activating certain circuits of our brain to make them more likely to fire in the future. You can think of how advertisement, for example, would take advantage of this type of priming to get your thought processes to go in certain ways when you see the advertisements. Another distinction in memory is the distinction in short-term and long-term memory. Short-term memory is like holding your friend's phone number in mind for a couple minutes while they run to a store. And it's often discarded and involves circuitry in the frontal lobes, keeping things active over time. A long-term memory, however, can last indefinitely, and it can last even our whole lifetime, and it involves activity in our temporal lobe. But there's not a single place in the nervous system where we can identify the location of a given memory, as we'll see in the course today. When we store memories, they're processed originally in the areas that they get processed in. For instance, when we see something, we process it in the visual area. When we hear something, we process it in the auditory area. And to some extent, the storage of memories also utilizes those same unimodal areas of our brain. So it's been shown, for instance, that when we recall colors, we activate the color sensing regions of our visual cortex. And when we recall words, we activate the word form visual area. Another aspect of our memories that's unique is we have these episodic memories in which we're actually a part of them. Episodic memories are also called autobiographical memories. These are for events that are at specific places and times that we are a main character in. There's even disorders called episodic amnesia, which are inabilities to recall experiences, but only your personal involvement in the experiences. So this patient, Casey, had a detailed memory of their life, of knowledge about life and how it worked, but not their own role in any of the events of their life. They had damage to their frontal lobe and reduced blood flow there. So it's thought that the frontal lobes may allow us to travel in our past and future in these ways and think about our role in our memories. Some humans have something called a highly superior autobiographical memory. These people can have virtually exact and complete recall for events in their lives, usually starting around age 10. They can describe any episode of their life, the day it happened, the day of the week it happened, sometimes the weather and other events that were going on. They've been shown to have increased gray matter in their temporal and parietal lobes and increased fiber projections between the two as well. Okay, next we're going to talk about the dissociation between implicit and explicit memory and how researchers first identified kind of the distinction. Researcher Carl Lashley was originally looking for the neural circuits underlying memories, and he trained rats to do a particular task where they held something in mind. And he cut different parts of their brain and found that the severity of the disturbance in memory was related more to how much of the brain they cut, not to the particular location. However, he was found to only have been cutting parts of the cortex, and so may have missed a very important area that was later identified to be important for memory. The Canadian part of the story comes later, but a surgeon in New York performed a bilateral medial temporal lobe resection on a young man named Henry Mollison, who was having uncontrollable epileptic seizures, and found that the seizures were originating in the amygdala and hippocampus bilaterally. And so they were removed bilaterally, and this is the first known patient to have bilateral removal of the hippocampus. A Canadian scientist named Brenda Milner had been studying patients that had unilateral hippocampal removal and had identified some memory problems in them. And so she was recruited to try to understand the memory disorders that HM experienced following the removal of his hippocampus and amygdala. Here you can see the lesion location. Here's the right underside of the HM's brain, showing this large area of both the amygdala and hippocampus that was removed. HM's brain was also sliced very thin and imaged for science, and you can view the images of HM's brain online. So following the surgery, HM and the surgeons were surprised to find that he had severe amnesia. He had no explicit memory of anything that happened new in his life. Couldn't recall anything that happened after the surgery. He had above average IQ, and he was perceptually fine, and he could recall events from his childhood. And even his performance on implicit memory tasks, like the tracing task or the motor pursuit task, or those pictures of degraded animals, was intact. Conversely, a different patient, JK, had impaired implicit memory while they had intact explicit memory. JK developed Parkinson's in their 70s and started to have memory problems at age 78. They were identified to have damage to their basal ganglia. They had an impaired ability to perform tasks that they had done all their life, like turning off the radio, but they could recall explicit events from their lives. So this is kind of a dissociation between this HM patient and JK patient, showing these two memory systems. So now we can go in and look at the neural anatomy of the two memory systems and think about how those connections give rise to these different types of memory. We'll see that explicit memory, as we might imagine, is generally subserved by areas in the medial temporal lobe, like the hippocampus and amygdala, but also these additional medial temporal lobe regions, the entorhinal, parahippocampal, and pararhinal cortex, which we'll learn about today, and also the frontal cortex. So the hippocampus and the amygdala are kind of tucked inside the temporal lobe here, but on the ventral surface of the temporal lobe, there's additional structures, including the pararhinal, parahippocampal, and entorhinal cortex. One can see that the incoming information to the medial temporal lobe comes in this funnel type direction from these cortical areas in through the pararhinal and parahippocampal cortices, through the entorhinal cortex, and ultimately to the hippocampus. And it's this circuit that seems to be specifically involved in explicit memory, not just the hippocampus itself. The pararhinal cortex receives connections from the ventral stream, the visual regions that take part in our object identification, whereas the parahippocampal cortex receives connections from the parietal cortex, the dorsal stream, and this is thought to be involved in visuospatial processing. So it's taking all this visual and spatial information and combining it at the entorhinal cortex. The entorhinal cortex is the area where cell death in Alzheimer's disease is first observed and shown to correlate with difficulty in explicit memory. The hippocampus and this medial temporal lobe system seems to be involved in more than just our explicit memory, but seems to have this visuospatial way of memorizing the world, in that it uses visual information to identify objects in their space. If you have an animal or a human with hippocampal injury, not only do they have these memory deficits, but they have very explicit spatial memory deficits. And monkeys with hippocampal lesions have difficulty learning locations of objects. So here's two very similar tasks. A monkey is trained first to lift up an object and to find food under it. And then upon completion of that first stage, they might have these two different tasks. One of them called a visual recognition task, asks the monkey by rewarding it to pick the new object in the second position. So the monkey has to remember that first object and then pick a new object. And monkeys with pararhinal or parahippocampal cortex damage seem to have deficits at this task. Whereas this very similar looking task in which an object is placed at a location, and then on the next task, the animal doesn't have to identify the visual form of the object, but the spatial location. In this case, animals with hippocampal damage specifically have deficits. So there seems to be different types of memory subserved by these different parts of the medial temporal lobe. One prediction is that animals that are good at spatial memory should have larger hippocampi, if this is true. And this does in fact obtain, and that birds and rodents that store food all over their environment have larger hippocampi than birds and rodents that don't. And so foraging and storing food seem to lead to larger hippocampal volume. And also it's been observed in humans that London taxi drivers who have this amazing spatial ability to get around one of the most complex cities in our planet have significantly larger regions of their hippocampi than control participants. Then this exciting study called the taxi cab study by a lady named Eleanor McGuire. And as the textbook even indicates, she went on to show that you can predict who's training to become a taxi driver, who will go on to pass the test based off the extent to which this area of their hippocampus grows during their training. So in support of this idea that the hippocampus has this very spatial role in our memory, one important clue is that there's neurons. If we look at the neurons in the hippocampus and we look at when they fire, they seem to have very spatial related activity patterns. There's these three types of cells in the hippocampus that seem to be related to like a global positioning system, a GPS system for our brains. There's things called place cells, which discharge when a rat is in a particular location, irrespective of which way it's facing. There's cells called head direction cells, which fire whenever a rat's head points a particular direction. And there's things called grid cells, which discharge at these regular grids in the environment, which can create like a spatial map of how far you've gone. So here's examples of those types of cells. These are showing different ways of plotting the activity of the cells. And so in this first one, A, they're showing when the rat goes to each of these locations in an environment, they're plotting how much the cell fires. And they find that this cell fires most in this one part of the circular environment. You can see that it doesn't matter which way the rat was going, any way they're headed, shown on this circular plot here, leads this cell to fire. Whereas there's a similar cell that fires in one location, but only when the animal's traveling in a particular orientation. There's also head direction cells, which fire generally throughout the environment, but only when the animal's pointing one way. And then these grid cells, which fire in these repetitive hexagon patterns, creating this spatial grid that the animal's brain can use to create a map. The researchers, a husband and wife named Moser and their former advisor John O'Keefe in 2014 won the Nobel Prize for this research, identifying this spatial positioning system in the brain. So there's reciprocal connections between the hippocampus and the medial temporal lobe and back to the cortex, and it's these connections between the hippocampus and back to the cortex that seem to keep our experience alive. If I want to remember something over an extended period, I can't just rely on the original activity. I have to create this circuit of activity that reactivates it every cycle. And the frontal lobe is important in this kind of circular activity where we keep things in mind in our short-term memory. So all of our sensory systems project here to our frontal lobe. We're not going to cover in this class the topics in chapter 15, which focus a little more on these frontal lobe tasks like short-term memory and attention. You can go on and take classes like Psych 375 in our department where we go in more detail on those topics. That's a class called Cognitive Neuroscience. And so during these tasks where you have to keep things in mind, like someone's phone number, there's cells in our frontal cortex that fire throughout those delays in which we're keeping things in mind. And if you don't remember the things, you have no delay activity in the frontal lobe. So our frontal lobe seems to be able to kind of keep our brain active in a particular state that we want to keep in mind. One important component of this explicit memory circuit is that it can get damaged by alcoholism. A disorder called Korskov Syndrome involves a permanent loss of the ability to take new information, like anterograde amnesia, and also to retrieve old information prior to the brain damage. It's caused by damage to the diencephalon, the thalamus, from chronic alcoholism and malnutrition. And both the alcoholism and the malnutrition together conspire to create a vitamin B1 or thiamine deficiency, because the alcoholism, I think it changes the ability to process thiamine in our diet. And then the diet can also be poor. But the symptoms look very similar to this kind of amnesia found from hippocampal lesions or amnesia from Alzheimer's. Retrograde amnesia refers to the inability to remember things that took place before the onset, and anterograde is an inability to remember things subsequent to the damage to the brain. The resection of the hippocampus leads to this retrograde amnesia, but not necessarily, it leads to anterograde amnesia, but not retrograde amnesia. So you continue to have the memories that you had prior to the damage to the hippocampus. This is one piece of evidence we'll talk about in this class, that our actual memories that are long-term end up distributed throughout our brain and aren't contained entirely in the hippocampus. This ability to retain information after resection of the hippocampus in the retrograde form. So these sensory and motor areas connect to the medial temporal lobe, and the temporal lobe structures connect back to the sensory areas. And so it's thought that like this connection between the temporal lobe and the cortex over time can solidify these long-term memories. And the connection between the prefrontal cortex and the temporal lobe is involved in keeping these short-term memories online, but also in thinking about the chronological order that things happen. So to bring this all together, here's this circuit for explicit memory. We have the incoming sensory and motor information into the cortex that funnels into the medial temporal lobe through those pathways we talked about. The medial temporal lobe has these reciprocal connections with the prefrontal cortex, and also with, and all of these areas receive activation from things like acetylcholine and noradrenaline from these reticular activating systems. But it's this reciprocal connection between the temporal lobe and the cortex, and then this reciprocal connection between the cortex and the temporal lobe that seems to be involved in here in short-term explicit memories and in long-term explicit memories. So the hippocampus seems to consolidate new memories over time. And as we've alluded to, they then move from the hippocampus to diffuse regions of the cortex. One of the best ways that I ever was explain this is that the hippocampus receives a memory like someone holding a bunch of strings with balloons, and the balloons all go to the various parts of the brain that contain that memory: the color red, the shape of a fire truck, the sound of the fire truck. The hippocampus is holding together all the various components of that memory and is required at the time. But then over time, as that memory is reactivated, those various components fire together and they begin to be connected together into a memory unit. And the holder of the strings can let go, and those memories can co-activate one another without the hippocampus being involved. This is thought of as the process of consolidation. And after consolidation occurs, then the hippocampus is no longer required. This relates to the idea of distributed reinstatement theory. So when we learn, we produce a stored memory that's strong in the hippocampus that has these connections to the other areas. But as it's replayed over time, multiple days and hours, the other areas' representations get strengthened and the hippocampus is no longer needed. Reconsolidation refers to the idea that when we activate a memory and recall it, we can reconsolidate, we can change it or stabilize it or weaken it. So whenever it's replayed, it's thought that we can further consolidate it. This is the idea of the movie with Jim Carrey called Eternal Sunshine of the Spotless Mind, in which he goes to a clinic to try to remove a memory from his brain. And they use a technique similar to this. They activate the memory and then they try to reconsolidate it so that it's no longer there. So the idea is that you could possibly erase negative memories when the memory is revisited by activating it and then changing the quality of the memory. Conversely to this explicit memory circuit, which we've covered, researchers like Mort Mishkin and colleagues have proposed that this is the explicit memory circuit, this temporal lobe with the frontal lobe and the thalamus, and the activating systems, which we saw in that previous diagram. I'll switch it now to the implicit memory circuit, which they propose, which involves a different set of structures, the basal ganglia, the ventral thalamus, and the substantia nigra, where dopamine is produced, as well as the premotor cortex, where our motor programs are created. The idea is that the basal ganglia receives input from the neocortex, and it sends information to the thalamus, and then on to the premotor cortex. Not only does it receive information from the neocortex, but it receives this distributed dopamine signal from the substantia nigra. And so this dopamine signal seems to be important for implicit memory because of the way that it activates the basal ganglia. So here we have the basal ganglia, which can output via the thalamus to make us move with our premotor cortex. So when you go to, say, ride a bike for the first time in a couple of years, you're exerting your behavior through this pathway. You're not explicitly remembering how to ride a bike. You're activating the basal ganglia and exerting that motor program implicitly. You can see the role that dopamine plays, as we know, in the basal ganglia function and how it might gate this implicit memory formation. Implicit memory is something we're not often aware of, and it's believed that this might be due to the direction of the connections between the basal ganglia, because the cortex doesn't reach back and reconnect to the basal ganglia like the reciprocal circuits of our explicit memory circuit. It's proposed that maybe we are less conscious of the activity in this circuit. This relates to this idea of the nature of consciousness, which we don't have time to cover in this class. It's also talked about in chapter 15. You can go on and take a class in this department about consciousness as well. We cover it a little bit in 375, and I've taught a 400 level consciousness seminar as well. But one of the main theories of how we might be conscious of something is by reciprocal connections within our brain, and when activity re-enters into a earlier area, that might create this phenomenon that we experience as consciousness. Thirdly, we have this additional type of memory, which we can think of as emotional memory. These are memory for the affective properties of a stimulus. These seem to be able to be implicit and explicit, and the amygdala is very important for this type of memory. So if you have damage to the amygdala, you can have no emotionality of your memories, but no difference in your implicit or explicit memory. As we can see, the amygdala has these close connections with the medial temporal lobe, which leads it to be very well situated to affect the memory circuits with its emotional properties. It also sends projections to the brainstem, where we can control our autonomic responses and heart rate and blood pressure, to the hypothalamus, which can produce hormones, to the paraqueductal gray, which can affect pain perception, and to the basal ganglia, where it can affect the implicit memory system. So this goes to show, yes, that emotional memories seem to be able to permeate through our body and affect our physiology, but also these additional memory systems that we've learned about today. And so the circuit of explicit memory seems to also be contained within this emotional memory circuit. We can see a quick diagram here showing the medial temporal cortex connecting to the amygdala, connecting to the basal ganglia, and all this information coming from the sensory areas like in the other models. The amygdala has this major role in adding this emotional twinge to our memories. And we know this because we tend to remember emotionally arousing things particularly vividly. it's thought that the neurochemical and hormonal systems that are activated during emotional situations can prime memory to be more solidified. And so a particularly happy or scary or stressful event can lead to a qualitative difference in the ability of our memory system to encode it. Okay, next we're going to talk about the structural basis, if we zoom in on some of these circuits, of why changes might occur over time in our brain. In general, the neural level, memory is associated with a change at the synapse. Because a synapse is where one neuron can influence another, any change in that influence can lead to a lasting change in the circuit. So the strategy to find correlates of memory in the brain is going to be to determine how synaptic changes correlate with memory. We want to localize the changes to specific pathways in the brain. We want to analyze the nature of the change at the synapse with a microscope and see what's changing. The most common way that's been used to study learning at the synapse has been called long-term potentiation, a form of associative learning. Associative learning is a linking together of unrelated stimuli to elicit a behavioral response. And long-term potentiation refers to the technique in which you stimulate the presynaptic neuron and you change the amplitude of the postsynaptic potential from a given input. And so you test how much the presynaptic neuron makes the postsynaptic neuron fire, and then you stimulate that circuit a whole bunch, and then you test it again how much the presynaptic neuron makes the postsynaptic neuron fire, and you find that it is much more effective at making the postsynaptic neuron fire after this long-term potentiation. So the stimulus in this case is artificial. It's a strong burst of electrical stimulation that you apply to the presynaptic neuron, but this leads to a lasting change in the amount of postsynaptic potential that's produced by a given presynaptic input. This was first recorded in the 1970s by sticking electrodes on the pre- and postsynaptic neuron of aplysia, a sea slug with particularly large axons. And in this case, they didn't just measure the EPSPs on a single neuron, but from a large group of neurons based off the field potentials. So here's an example showing the stimulation and the recording. So here they stimulate and record the amplitude over and over and over and over again. Then they do this large stimulation period, following which, when they stimulate and record the amplitude, they find a much larger EPSP. The stimulation was just a very rhythmic, high frequency electrical stimulation. And so what's happened here is the synaptic connection between the pre and postsynaptic neuron has changed, and now a given input from the presynaptic neuron is more likely to make the postsynaptic neuron fire. Or the synaptic weight has changed. And for this EPSP to increase, what this means is that there's either more neurotransmitter released from the presynaptic neuron, maybe the postsynaptic neuron is more sensitive to that neurotransmitter, or both. We can have the opposite effect, long-term depression, a decrease in the EPSP size. Perhaps this long-term depression, therefore, is a way of clearing out old memories. The way that you elicit long-term depression is during this stimulation period, instead of electrically stimulating 100 times a second, you stimulate like 5 times a second at a slow sleep-like frequency. And this leads to a lasting decrease in the amount of postsynaptic potential. Another important synaptic mechanism by which learning occurs is through this change in connectivity caused by activity. You can think back to this idea that neurons that fire together wire together. This particular circuit and set of mechanisms that was observed, this AMPA and NIMDA glutamate gates on the postsynaptic neuron is one way of explaining how when two neurons begin to fire together, they increase their ability to connect and wire together more likely. So how this works is that glutamate, when it gets released by an excitatory neuron, can act on two different types of postsynaptic membrane receptors. It can act on AMPA receptors, which are normal. They just respond to glutamate and change the postsynaptic potential, and also NIMDA receptors, NMDA receptors. These are double gated channels, and they're normally blocked by this large magnesium ion, and so they don't respond to glutamate. And it's only when something occurs can this magnesium ion move and allow the NMDA receptors to respond to the glutamate. And it's specifically when the presynaptic and the postsynaptic neuron are already both firing that this will occur. Let's see how this occurs. So the magnesium ion moves away from the pore whenever there's a strong postsynaptic membrane change. And so if the postsynaptic neuron gets activated and it changes its potential, it can then release this magnesium from its NMDA pores, opening up those receptors for even more activation by glutamate. And so we'll see on the next slide, and two here. Let me go to the picture because it shows it much better. So at first, a very weak electrical stimulation is applied to the presynaptic neuron. And because the magnesium pore is blocking, the glutamate only attaches to this AMPA receptor and creates a small change in the postsynaptic potential. If enough change in the postsynaptic potential occurs through these AMPA receptors, the postsynaptic membrane can become depolarized to the point where this magnesium ion gets released from the NMDA receptor. And now the NMDA receptor is open for glutamate to bind to it, and calcium gates open, and calcium can come in the cell and create a second messenger cascade. For instance, that one of the things it can do is create more AMPA receptors. And now we can imagine what that would do. More AMPA receptors would make the postsynaptic neuron more likely to fire when a given presynaptic neural input occurs. And so by identifying a period where both the presynaptic and the postsynaptic neuron were both active and only changing the synapse during that time, this mechanism allows for neurons that fire together to begin to wire together slowly over time. When I first learned about these NMDA receptors as this mechanism for increasing synaptic connections, specifically in circuits where the presynaptic neuron made that postsynaptic neuron fire, it was very exciting for me, I recall. I'm going to go back to cover those slides again. So just to cover that again, two events have to occur for the NMDA receptors to open. It has to be a depolarization of the postsynaptic neuron, so the magnesium leaves, and glutamate still needs to be released from the presynaptic neuron, even if it's just a weak stimulation, but enough to activate those NMDA receptors. So the strong electrical stimulus that makes the magnesium pore leave and the weak electrical stimulus that makes the NMDA receptor active are now paired. Maybe you can see this is also a mechanism by which something like the activity from a bell, which might only weakly activate a circuit, could come to be paired with the activity from something like a food, which creates a large activity in the circuit, such that slowly over time, the bell comes to make that circuit fire more because of this repeated pairing of the two stimuli together. After the NMDA receptors open, we've seen that then the calcium enters, and this can alter the postsynaptic neuron's function. We saw that it can increase the number of AMPA receptors, it can create new AMP receptors. It can even send retrograde messengers to ask for more glutamate to be released from the presynaptic neuron. Interestingly, long-term potentiation has even been shown to occur on GABA interneurons, which release inhibitory GABA. So there's a similar phenomenon to LTP and LTD called LTPI and LTDI. These are inhibitory long-term potentiation. So these inhibitory neurons can also change over time through experience. So when we think about long-term learning, we can think about the calcium entering and changing the activity of the postsynaptic neuron. Further thing that happens is the cyclic AMP is involved in that second messenger cascade. And this can alter the gene expression of the postsynaptic neuron, which can physically alter the synapses themselves. So there will be structural changes to the dendrites and synapses and an increase or decrease of synapses. So the actual structure of the neurons is going to change, not just their number of receptors or the receptor inactivation. There could be more dendrites. And this is one way that researchers infer synaptic changes. They look at the dendrites under a microscope. And these new synapses can either form between neurons that were already connected that strengthen their connection, or even between neurons that weren't previously connected to create new connections. Here's some examples of that dendritic plasticity looked under a microscope, showing the same neurons after 90 days of enhanced environment, showing this increased dendritic growth. And researchers have shown, for example, that if you raise young rats in a very enriched environment with lots of other rats, that after that experience of enrichment, they can have increased number of synapses and increased synaptic density such that there's new axons from new neurons or new collaterals from existing neurons, and various different dendritic spine shapes that grow. Prior to the 1990s, it was generally thought that our mammalian brains don't make new neurons, but there's evidence now that new neurons are constantly being generated in the olfactory bulb and the hippocampus and maybe even our neocortex. It's unclear why new neurons are being generated. But we know that this neuronal growth might be an important aspect of learning as well. As I talked about, we can study learning through a number of ways, and one of them is we can change the environment in which rats or animals grow up in. So if we raise rats in these really enriched environments with lots of toys and other friends, we can find that we increase their brain weight, have more dendrites and astrocytes, more blood flow to their brain, more synapses, and increase mitochondria, the power stations of the cell. So indicating that they have better metabolic activity as well. And so there's this amount of almost 50% increase in vascular volume and 10% increase in dendritic length just from raising rats that would normally be raised in a very boring kind of pet store type environment to a fun cage with lots of other friends. And this has led to policy decisions in the United States, for example, of the Head Start program, in which there's a lot of resources dedicated to identifying individuals that might benefit from early interventions in kind of preschool and kindergarten to try to counter the issues associated with low socioeconomic status. Some other examples of experimental plasticity were done in the 80s by a researcher named Bill Greeno, where he placed patches over one of the eyes of a rat. And because rats' visual systems are largely completely separated. That is, their right eye almost entirely goes to their left hemisphere, and left hemisphere almost entirely goes to their right eye. They were able to show that the visual cortex of the trained hemisphere, the eye that was open, had more extensive dendrites after a couple of weeks of training on a maze with only one eye. Even more specifically, these colleagues in the 90s showed that if you trained monkeys to retrieve food out of a well, either using very slight movements of their fingers with a tiny well, or big movements of their hand with a larger well, they were able to show that the motor representation of the fingers was much larger following this training in the difficult tasks than in the easy tasks. So depending on the activity we do throughout the day, our brain can change, and areas of our brain can come to represent parts of our body that we're using more. Relatedly, in humans, Ramachandran showed that and inferred that individuals that have a limb amputation have adjacent areas in their cortex to the area that represented that limb that might come to represent that limb, because there's no longer information coming from that part of the body. So now we have a part of our brain here which used to represent the hand. It's no longer receiving information from the hand. Slowly, the area adjacent to the hand, which is the face, the neurons that come from our skin of our face, might slowly branch out and connect to those neurons that used to be associated with the hand and come to activate them. And Ramachandran found that when the face was stroked with a cotton swab of these patients, they reported sensations in their hand, which he inferred indicated that the representation of their homunculus here had kind of morphed such that now their face, incoming information from their face, was activating regions that they were still attributing to their hand. It's even shown that because we use certain parts of our body for more complex things, the dendrites of the neurons that represent those parts of the body come to be more dense than the dendrites that represent parts of our body that do less complex things. So the neurons that receive input from our fingers have very intense dendritic branches versus those that come from our chest. The somatosensory neurons that come from our chest have much smaller dendritic branching. And furthermore, in studies that they did of workers after they had passed away, they found that people that were word processors on a computer had large differences between their finger and their trunk neurons, dendrites, whereas salespeople in kind of 1980s sales stores with no computers had very similar representations between their finger and trunk. So using your hands all day to enter text on a keyboard is altering the neurons physiology that represents those parts of your body. So here we can see more dendritic branching in the finger area and the language area than in the trunk area, which doesn't have much complexity in its task. Finally, the epigenetic influence of memory seems to indicate that we can actually change the genetic coding and expression in certain parts of our genome, and this can affect our memory. So, for instance, fear conditioning can be associated with rapid methylation of the DNA. And if you block the methylation of the DNA with chemistry, then there's no memory created in this case. And just to recall, methylation of a gene of DNA can affect its ability to be expressed. And it's thought that some of these epigenetic mechanisms might even be passed on to multiple generations. So plasticity can also be influenced by things like hormones and drugs, which we've seen a little bit throughout this class. We saw that high and low levels of estrogen can affect the amount of dendritic spines in the hippocampus. This might be related to memory decline in menopause. And low levels of testosterone can can lead to more dendritic spines in the neocortex. And this might be related to a decrease in spatial ability as men age and lose their basal testosterone levels. Glucocorticoids are released from the adrenal cortex in times of stress. This is like the adrenaline response when a very stressful thing happens. These provide assistance to our metabolism to help us get energy from the food when we're in these stressful situations. However, steadily over time, exposure to these stress hormones can lead to neurotoxicity. We've seen that you can lose hippocampal cells with chronic stress. And we even saw in the chapter that these hippocampal cells seem to be important for modulating stress, and a vicious cycle can occur in which the stress can then spiral out of control. We have a number of neurotrophic factors in our body that can affect nerve growth, and important research is focusing on these as well. So nerve growth factor can stimulate neurons to grow new dendrites. And BDNF, brain-derived neurotrophic factor, is another chemical that our body makes. And we can use it possibly to enhance plasticity and the growth of dendrites. And when you have animals learn to solve a maze or a problem, you find increasing levels of BDNF in their blood following that learning experience, indicating that these might be kind of some of the messengers that lead to long-term synaptic changes. Drugs can also change neural circuits. So there's something called behavioral sensitization. If you take repeated administration of a drug like cocaine or amphetamine, you can have an increased behavioral response over time to that drug, especially if the drug taking experiences are spaced out apart in time. This seems to be associated with increasing number of receptors and synapses and dendrites in the neurons in microscope images. And these changes are generally located in regions. The synaptic changes are preferentially located in regions like the prefrontal and nucleus accumbens, which receive large dopaminergic projections. So it's thought that the interference with the dopamine system by these drugs can lead to these kind of synaptic changes that may be aberrant and lead to possibly negative drug-taking behavior. The textbook lists these kind of 7 principles of brain plasticity, which are good to think about as well. Any behavioral change reflects some brain change. All nervous systems are plastic. Plastic changes are age-specific. Can't teach an old dog new tricks. Prenatal events can influence brain plasticity. Plastic changes are brain region dependent, so that you're not going to have plasticity throughout your brain, but on the circuits that you're using. And experience-dependent changes interact so that you can have both like a change from learning, but also a propensity to have that change from your past experience. And it has pros and cons. There's good things and bad things. 1 is that we can learn, and 2 is that we can get into these learning ruts. Finally, we're going to talk about the recovery from brain injury. So the textbook talks about Donna, who had a traumatic brain injury where she did show some recovery, but it wasn't complete. There were still things that she couldn't do. She had a brain network that was damaged, and she had a rest of her brain that could then maybe take over and try to cover up some of the lost function, but nonetheless, there's still this damaged circuit. There's 3 possible ways that we can think about recovery from brain injury, and we can research new techniques to try to help people recover. 1 is to learn new ways to solve the problems, to use existing tools and circuits to solve problems that are caused by the lack of the damaged ones. The brain can reorganize in ways to do more with less that it has now. And the brain can also generate new neurons that can produce new circuits. And this is the most exciting kind of avenue that this research is going. So the first of the three-legged cat solution, is that when a cat loses a leg, over time it can quickly compensate. It doesn't grow a new leg, but it learns how to walk on 3 legs. So a human that loses an ability, like maybe their memory, might first try to compensate by writing things down or keeping a notebook. And someone that might lose ability in their right hand might compensate with their left hand. A more high-level change after brain injury that might help is this creation of new circuits. So in response to an injury, the brain can make new connections. And these amount of connections and new connections are enhanced when the individual does additional therapies like speech or physiotherapy, or a pharmacological intervention like taking amphetamine or nerve growth factor. The ability of these new circuits to be created and strengthened requires these kind of additional engagements of the circuits. And finally, the one promising solution for fixing damaged brain is the ability to implant new neurons into an area that can then distinguish themselves and attach to the network and replace the missing circuits. One technique is to use tissue implantation from aborted fetal tissue. There's very limited success in this domain, and we've learned about one in the chapter on sleep, for instance, in which the neurons from the suprachiasmatic nucleus are injected into a rat that is not producing any rhythmicity in their suprachiasmatic nucleus, and that these neurons were able to invoke a rhythm. And some indication that you can take dopamine producing cells and you can implant them in the substantia nigra of Parkinson's patients. Although there's ethical concerns with the source of the tissue, of course. Alternatively, we can try to encourage the creation of neurons by our own physiology. So there's a growth factor called epidermal growth factor that can stimulate the subventricular zones around the ventricles to generate new cells, new neurons that then can migrate to the striatum and differentiate into neurons. And so maybe you could use some of these factors to create lots of neurons and have them migrate to a place of injury. The textbook chapter ends on this amazing image showing a lesion of a cortex of an animal. And then after this treatment with the nerve growth factor, in which new neurons are created and they migrate to the area of the lesion, it looks like the brain has been kind of rebuilt in that area. Although the textbook warns that this part of the brain was not integrated in with the rest of the brain, and only slightly improved the behavioral function, although this does seem like a very promising direction. So today, this brings us to a close. We've talked about learning and memory in general. We've dissociated implicit and explicit memory, and we've talked about the neural circuits underlying them, as well as emotional memory. We've talked about long-term potentiation and AMPA and MMPA receptors and how they might lead to changes in synaptic effectiveness and also synaptic physiology. And we've talked about various ways that we might try to improve recovery from brain injury using new circuits or novel generation of neurons. Thanks very much.